Hi, my name is Stephanie Monteith, and for the purpose of this video today, we're going to be talking about gram stains and the difference between gram-positive and gram-negative bacteria. So the difference between a gram-negative and gram-positive bacteria. First off, in a gram-positive bacteria, you have a thicker layer of peptidoglycan. So the thickness in the layers that actually surround the plasma membrane is kind of what really differentiates between the two. So to be able to see this under a microscope, we do a process called a gram stain, and it's a 4-step process. Starting with step 1 is the staining of the crystal violet. The crystal violet stains all the bacteria on the slide to a purple color. So we let this sit on the slide for a minute. Once we've allowed that to sit for a minute, then we rinse it off. Like I said, during this step, all the bacteria on the slide is now a purple color. So we then move on to our second step, which is the iodine. And in this step, the iodine is going to help fixate the purple color in the gram-positives. It allows it to bind to those cell membranes. So we also let this stain sit for 1 minute, and then we rinse it with water. Our third step is the decolorization step. The decolorization is done with a alcohol or an acetone. It's a lot quicker than the first 2 steps. It's only about a 5-10 second step. We just splash it on and splash it off and rinse it with water. We don't want to leave it on too long because it will decolorize all of our slides or all of our bacteria. So at this point in our stain, we have a set of gram-positive bacteria that is stained purple, and we have gram-negative bacteria that is colorless at this point. So if we were to look at it under a microscope right now, all we would see is purple and colorless gram-negatives, so you wouldn't really see them. So our fourth step is going to be the saffron, and that dye is what's going to help allow us to see the gram-negatives under a microscope. So for this step, we also submerge the top of our slide with the stain, and we allow it to sit for 1 minute. After we let it sit for a minute, then we rinse, and then we can blot dry our slide, and then we can use oil and we can look at it under our microscope. So you'll have 2 results. You have the gram-positive bacteria that's showing in a purple color, and your gram-negative bacteria that's going to be shown in a pink color. So a lot of times, especially in different healthcare facilities and clinics and things like that, you'll use a gram stain to kind of show what bacteria is going on, and whether or not you can use antibiotics, or if you can see things like yeast and different things, organisms on your slide, and they use these in hospitals and situations to help kind of further diagnose and to know whether or not the treatment's going to work. For example, in the clinic that I work at, we do a lot of gram stains for fecals, and we diagnose patients all the time with Clostridium and yeast. We can see blood in the stool. We can see all sorts of things along with these gram-negatives and gram-positives. So yeah, that's the process on gram staining, and I hope you enjoy.